yeah, with this, we have an excellent speaker to open our series of web seminars. So we got Dr. Li. So he got his bachelor degree in biosciences from University of Science and Technology of China. Then he obtained his PhD in computer science from CASP in Saudi Arabia in 2020. But the same year, in December, he joined the Department of Computer Science and Engineering at the Chinese University of Hong Kong as an assistant professor, leading the Artificial Intelligence and Healthcare Group. He's also visiting assistant professor at MIT and Harvard, working with Professor James Collins. I hope we'll see each other next week. I will use this opportunity to network. In 2022, he was selected to the first 30 under 30 Asia list in healthcare and science. He works at the intersection between machine learning, healthcare, and bioinformatics, developing new machine learning methods to solve the computational problems in biology and healthcare. And he was involved in a group that you may already know that performed very, very well and outperformed other groups in the recent CASP RNA experiment. What was particularly interesting that the group proposed two approaches, classical ones based on simple sample-based methods and deep learning one. And actually, it was very surprising to see that the classical method outperformed the other one and also the other groups. So I hope today we'll learn more about that. I hope we'll understand more contribution of each groups. And also, I think, hopefully we'll also explain better the tools that are produced by the group. And yeah, with that, I'm super happy to be here. And just a reminder, after this, we'll have discussion for the community about this initiative, and we can discuss in which direction you would like to take this initiative. But yeah, with that, Professor Li, the screen is yours.
Yeah, so I was here. So I was discussing that for the RNA, similar to protein, we have the primary sequence, secondary structure, 3D structure, and function. So we all know that the primary sequence will first fold into the kind of base pairing to fold into the secondary structure, and then we'll further fold into 3D structure. With the 3D structure, they can interact with the other molecule to show some function. So we know that for regarding the bottleneck of RNA studies, we know that the RNA are relatively more flexible than the protein, and so by now we still have the data scarcity issue for the RNA 3D structure. So by May 2022, in the PDB database for the protein, we have roughly 185K structures, while for the RNA, we only have 5K. So that's also include the protein RNA complex. So if we remove the redundancy, we probably only have around 1,000 RNAs with the 3D structure. On the other hand, in addition to this data scarcity, we also have the unknown function for the RNAs, especially for the non-coding RNAs. So we also lack the annotation for those non-coding RNAs. On the other hand, the RNAs are very important and abundant. So think of it, if we can develop some drug that targeting against the RNAs, probably will have 100 times more targets than protein, because some of the RNAs won't be translated into proteins. For the largest database that we can use for the non-coding RNAs, RNA Central, there are roughly 27 million sequences. So in our mind, in our group, we want to try to take advantage of such huge amount of unannotated RNA sequences to try to promote the development of models to predict the RNA structure and function. So based on that, we developed the RNA language model. We call it RNA foundation model, or RNA-FM. So essentially, this model is very similar to the language model in the natural language field. So do you have any question here? Okay, yeah, no question. So essentially, this model trained similarly as the natural language. So we try to extract information from the larger amount of unannotated data, and we train this model trained on all the RNA central non-coding RNA sequences using self-supervised learning. That is, for the RNA sequence, we'll try to mask part of the sequence, and then using the left chunk of sequence to predict this masked one. So after trained, the model can output the representation of input sequences, and we show that it can benefit all the downstream structure and the function prediction applications. So I'm going to show you how this model can improve the prediction of RNA structure as well as the RNA function. But before that, I want to show you what has this model learned from the sequence. So essentially, here are some visualization of the embedding, that is the representation of the RNA sequence using t-SNE. So essentially, we show that the RNA foundation model can extract the sequence pattern and also the evolutionary information from larger amount of sequence. That is, we have an assumption that by considering all the RNA sequences, this model can learn the language of RNA, which contains the sequence pattern as well as the evolutionary information. After checking these embeddings, we want to show that such embeddings can help us to improve the performance of RNA segment structure prediction, 3D structure prediction, as well as the final downstream function prediction. So let's check them one by one. So for the segment structure prediction, the problem formulation is like this. So given the primary sequence, we want to predict the base pairing within this RNA sequence, or we can represent either using a graph or using a matrix. So within this matrix, if two base form a base pairing, then we will put one into this very sparse matrix. So this is essentially the problem formulation. So traditionally, what people usually do is try to consider each pair has a certain energy, and then we're going to optimize over the summarized energy of all the base pairs. So this is a traditional method. And we can say that there is a very suitable algorithm for this kind of problem, that is the dynamic programming. And in fact, before our method, so all the previous methods were based on dynamic programming. So we can say this method was roughly from 40 years ago. And this one, RNA-FM, which is also another famous method, was only published a few years ago, still based on dynamic programming. But some problems here, like dynamic programming is slow and performance improvement over the past 35 years is not very significant. And the dynamic programming is standard and unable to handle the nodes. So essentially, we try to resolve this problem from another aspect. That is, we try to consider this as a translation problem instead of an energy minimization problem. So we propose a deep learning method to consider it just like a translation, right? You can translate this sequence into this matrix. Okay, so we can also incorporate some prior knowledge into this model to constrain this searching space. Okay, so with that, we propose the model called E2E-fold. So essentially, this is a kind of deep learning method to translate it into the final content map. And this content map has a one-to-one mapping against the segment structure. Okay, so I mean, I understand that most of your interests are in the 3D part, so I'll just go through this part very quickly. And this is the result. So at that time, roughly 3 years ago, we can improve it a lot. But later on, we find that this model has some overfitting issue. That is, it can only predict the family that is within the training data. So essentially, we try to see whether this RNA foundation model can help us resolve this overfitting issue and try to extend the prediction to the unseen families. We found that with this RNA foundation model, even if we just modify it in a very simple way, we can already outperform all the previous methods, including this RhoFold, which was published early 2022. So we also have some experiments, although I didn't put the slides here because the work hasn't been published yet. And we found that this RNA foundation model's prediction can indeed extend to the unseen RNA families. Okay, so this is for the secondary structure prediction part. Then I'm going to discuss the 3D prediction part using more time. Okay, so I assume that for the audience here, you definitely know the RNA 3D structure prediction very well. So essentially, we try to predict the 3D coordinates of the atoms within the RNA and compare with the ground truth. So usually we would compare with the crystal structure obtained from the experiments, although we know that sometimes the crystal structure may not be that accurate. Mainly because the RNA is dynamic and it can have multiple conformation. But for now, we will usually, yes, this structure is an experimental artifact. That's exactly what I want to say. Yeah. So, but for now, sometimes we will consider the structure within the PDB database as the ground truth. Yeah. Again, so for the bottleneck, performance is still not as satisfactory as the protein field. Yes, exactly. I totally agree with you. So you can here actually it should form into a helix. Yeah, exactly. So for the we have the data scarcity issue, and if we use the traditional methods like the simulation and the physical-based methods, we usually need to do the larger scale sampling. It can be time-consuming and computational demanding. So we aim at develop an end-to-end deep learning method that can hopefully give us some reasonable prediction in a very, so hopefully can also be very quick. So previously, we know that there are roughly 3 type of methods. So either is template-based modeling, or the RNA fragment assembling and folding, and also the deep learning-based scoring function, the ARIS, developed by Rachel Das group. So for the template-based method, currently because the result of the RNA structure is very limited, so the template library size is quite limited. And for the RNA fragment assembly and folding, so the largest scale assembly can be computational demanding. For this scoring, so for this deep learning based scoring function, again, we still need to have the candidate 3D structure. So regarding our method, the intuition behind our method, so now we call it a RhoFold. Previously, we call it E2E-fold 3D. So the intuition behind it is that we want to do end-to-end prediction from the sequence to the wider structure based on deep learning directly, so that hopefully we can take full advantage of the training data and also speed up the prediction. So we also want to do some in silicon data augmentation and additional database using additional database information so that we can extract more additional information and also elevate the overfitting issue and avoid only remembering training data. So we also want to force the model to learn the folding principle instead of only remember data. So we tried to incorporate the expert's knowledge and constraints. So with this intuition and the design principle, we propose this method. In fact, this method is very similar to RhoFold, but because RNA is still different from protein, so we have some well-customized module for the RNA structure prediction. So essentially, given the RNA sequence, it will first go through a database to construct the RNA multiple sequence alignment. And then we will combine this multiple sequence alignment with the previous RNA foundation model to try to extract more pattern as well as evolutionary information from the multiple sequence alignment as well as the original sequence. And then those embeddings, both the pairwise embedding and the multiple sequence embedding will go through the RhoFold. This RhoFold contains 12 layers to further extract the information. You can consider this embedding here contains lots of the evolutionary information and the pattern from the database. Then it will go through the structure module to try to convert the information into the coordinate of the atoms within the RNA. So this structure module is essentially designed based on the knowledge of the expertise for this RNA field. In the final module, we try to construct the RNA structure. In addition to given the 4-atom 3D coordinate of the atoms, we will also try to predict the confidence of the prediction. That is, in addition to given the coordinates, we can also show whether our model is confident into the prediction or not. So this is the per atom confidence. So even for just one structure, some of the regions we can tell that probably this prediction may not be that accurate, while for the other regions, probably the prediction we are very confident regarding it. So when we train this model, we also incorporate a number of knowledge, including the sampling structure, the local frame and torsion angle, as well as base pairing constraints in the 3D space. Okay, so this is essentially the RhoFold system design. To check whether this model performs well on the RNA 3D structure prediction, we tried to perform very comprehensive evaluation. So we collect all the RNA structure data from the PDB database. We perform some necessary filtering. Then we try to do the evaluation from multiple different aspects, including using the standard computational RNA structures, using the tenfold cross-fold validation, and also using the cross-family and even cross-type validation. Finally, we want to show that the prediction from RhoFold can potentially be helpful to try to help the construction engineering to hopefully resolve more RNA structures. But of course, for this part, we haven't done any wet experiment yet. Yeah. So let's try to check the results here. So for the RNA puzzle targets, we first check the performance here. So essentially, we can say if we don't consider the RhoFold, we can definitely say FAPA2 and ARIS essentially give us the best performance across almost all the RNA puzzle targets. Yes, so FAPA2 and ARIS are indeed very good. For RhoFold, so for most of the structures, RhoFold can perform well. But for this one, puzzle 24, the performance of RhoFold is not that good. And we try to check the reason of this performance, and we found that here there is a pseudo node within the structure. So if we don't know the segment structure, so the prediction can be sometimes can be not very good. But if we know the pseudo node over there, the performance can be quite well. Yeah. And in addition to do this kind of blind test, we also want to check whether there are some overfitting. That is whether performance is just because we remember data, or whether it's because it can indeed learn some, at least learn some folding principle. So we check. So in this figure C, we try to check the performance against the maximum sequence similarity against the training sequence. That is, we check if the sequence similarity, the maximum training sequence similarity in the training data against the target is as low as 0.5, whether the performance is still okay. So we find that for some of those RNA targets, the performance is still quite good. And we also try to check the running time of our model. We find out that RhoFold itself, if we already construct the multiple sequence alignment, the running speed is like we can predict the structure in almost 1 second. But we have to admit that for the RNA multiple sequence alignment construction is still relatively time consuming. And we are working on that to improve the speed of construction the multiple sequence alignment. Yeah, and here are some ablation study. For this CASP 15 blind test, we're going to discuss it more later. Yeah. Oh, yeah, here it is. So essentially, here are the performance of RhoFold on the 4 nature RNA in the CASP 15. I would say for this one, the performance is not very good. For the other one, they are essentially very close to the best performance of all the other methods. So this is essentially what we get. And I would say for the nature RNA, RhoFold performance is quite okay. So what RhoFold didn't perform well is in fact for this human designed RNA. I discussed with some experts, and our kind of insight is that we found that this RNA designed RNA, their pattern is somehow very easy to be, I would say, to be known by some experts. So, you know, this human designed RNA targets this year are somehow biased to the manual prediction. And we have to admit RhoFold is not that intelligent enough to handle this very long and, you know, this kind of RNA targets. Yeah. And we further check the performance of RhoFold on all the RNA structures within PDB. And here is the detailed performance of each RNA target. So essentially here we perform the cross-fold validation. And we can say, so essentially the performance is mostly okay for most of the RNA. But if the sequence length is longer, then the performance is not very good. So there are multiple reasons for that. One reason we can think of is that, in fact, we don't have a lot of long RNA sequence available in the PDB database. So the training is, in fact, not enough to handle those long sequences. So I believe if in the future we can have more longer RNA structure within the database, I believe this RhoFold will perform much better for those long RNA sequences. Yeah. Here again, we try to check whether there's some overfitting issue, and if the sequence similarity is low, whether performance drops a lot, and we find out that's not the case, and so there's no serious overfitting issue over here. Yeah, and there are some case study further to show if we try to extract the most similar sequence of the RNA against the target from the training data, what would it be? So here, this purple one represents the structure prediction from RhoFold. This pink one is the experimental one, and this most similar one is, I don't know, probably it's blue, right? Shows the structure of the most similar RNA sequence from the training data. So we can say we didn't just extract the most similar sequence. Instead, we try to predict the structure. Yeah, but of course, we admit for some of the targets, it didn't perform well yet. And yeah, so here is more discussion about the RNAs of different lengths. So essentially, most of RNAs with 3 structures are shorter than 150 nucleotides. So RhoFold performs relatively consistently across different lengths if the sequence length is shorter than 125 nucleotides. So it may fail on the very long RNA sequence, potentially due to inadequate training on the larger RNAs. Yeah. So here we try to challenge RhoFold using the cross-type and cross-family validation. That is, we want to check if the RhoFold's prediction can be extended to the unseen families. So which means if we say that a family will be our testing family, then all the RNA sequence and structures won't appear in the training data. So we can say for most of the structures, so this is a kind of a random splitter. So for most of the RNA families, the performance is okay. While for some RNA families where the miss its performance is not good yet. Yeah. We also further challenge the prediction of our RhoFold using the cross-type. So, you know, cross-type is an even more difficult task. And we found that compared to the cross-family, the performance indeed dropped a lot. So we try to benchmark our method against the FAFA2 method. We find that indeed the performance improvement is not that much, but still we are comparable and sometimes better than FAFA2. even on this cross-type prediction. Yeah. And here I actually want to discuss this one. So as some of you have already pointed out, so in the PDB database, this is an artifact. So the ground truth is in fact not the ground truth. So there should be a helix here. And the pink one is the structure from the PDB database. And this, how to say, probably it's blue one, right? It's the prediction from RhoFold. And in fact, we found out that the prediction from RhoFold can reconstruct the biological relevant structure correctly, not to predict the structure as the artificial, the artifact in the PDB database. Yeah. So from this one, we guess that RhoFold indeed learns some folding principle instead of just remember database. Yeah. And here is another experiment. This is not a kind of prediction. This is in fact a retrospective study. We want to show that RhoFold can be helpful to resolve more RNA-3 structures. So essentially, we try to check whether the RhoFold can predict the interhelical angle. Yeah, we have some question here. There are 3 targets have very good structure template. What about the performance of RhoFold on another 5 targets? Okay, this is a very good question. And in fact, we have performed a very detailed analysis on all the RNA structures. But because the paper hasn't been published yet, so we will be able to soon see the result of all the RNA targets in the CASP 15 targets. Yeah. Thank you for the question. Okay. So for the okay, so I'm going to continue here. So for this experiment, we want to check whether RhoFold can predict the interhelical angle within the structure. You know, this helical angle can help us to do the trimer and dimer design. So essentially, we find that the prediction from RhoFold is very close to the even no matter it's cryo-EM experiment or the AMR experiment. Yeah. Okay, so it's already 40 minutes. I will try to speed up. Yeah, so this is the part for the 3D extraction part. I don't know that you probably have lots of questions. I will try to finish this function part very quickly so that we have some time for the Q&A session. So for the function part, we essentially want to check the deep learning methods, the performance of these methods to predict the protein RNA interaction. So we know for the protein RNA interaction, essentially there are 2 kinds of experimental methods. That is, we can try to determine the structure snapshot of RBP and RNA complex, or we can use the in vivo assay or in vitro assay to check the binding preference. So such as the RNA compete and selects. So for the previous methods, they are essentially sequence of assay-based methods. So we are trying to predict either the binding preference if we have the assay data, or try to predict it's a kind of binary prediction, say whether it can bind or not. So essentially, for obstacles of using the structure data, so there are multiple binding pockets and rotation can be quite complicated. So for our idea, we want to scan the entire protein surface and also extract some rotational environment feature to try to see if we can predict the binding landscape on the entire protein surface. So with that, we propose the nucleic net. So this is a binding. This is a deep learning framework can predict binding specificity of our constant at any location on the protein surface. So due to the limit time, I won't go through the details here. So the paper has already been published. Notice that this paper was published before RhoFold was proposed. So at that time, we only have very limited data. But now we're thinking of trying to extend it, considering the RhoFold as well as the recent advance in the RNA field. Yeah. So I'm not going to go into detail here because it's actually not related to what they are interested in. So yeah, so essentially cross-fold validation and also validated with the in vivo assay data. So so you know the theme of the here. So we want to say whether the RNA foundation model can be used to improve the performance. We found that with the RNA foundation model embeddings, we can reach similar performance as the methods using the real signal structure profile. Yeah. Okay, so here are all the softwares as well as code tutorial and servers. So all of them are publicly available. So feel free to check them and feel free to raise any questions if you have any, or if any suggestions to improve those methods, feel free to let us know. So I would say all those methods and works won't be a success with the help of all the collaborators here. So I want to thank all the collaborators here. And finally, yeah, for the acknowledgement. Yeah, I mean, this is not related. Yeah, just some postdoc in the PhD position, so valuable in our group. And thank you. Yeah, so I'm more than happy to take any questions you have.
Okay, so yeah, we are ready for questions. So yeah, maybe with Janusz.
Yes, this is super cool. I really like your talk. And yeah, thanks for responding quickly during the talk to my comment on these experimental artifacts. Actually, we discussed it today during our journal club. And we believe that many of such things can be actually real, but they are experimental artifacts because of the experimental conditions. One is the concentration dependence. So some of the just because depending on the concentration. And this is one of the factors that is not taken into account, right? You're not predicting the RNA structure given its concentration. And the second, which is most important, and this actually is a part of my question and the suggestion. Many of these RNAs that have been determined and deposited in the PDB are not just complete molecules, but fragments of bigger molecules. Yeah. And in fact, exactly. For instance, this riboswitch is only a 5' domain within an RNA that can be super long. Of course, it's most likely folded quite independently, but there is a big difference between the 5' end and the 3' end. In reality, the 5' end is actually the end, and in bacteria, it has the triphosphate, not the monophosphate, probably. But then the 3' end extends further. I think it would be interesting to include, I mean, this is a comment to everybody. I think it will be important to include the consideration of ends in the prediction of the RNA, whether there is something beyond the 3 terminus, for example, or whether there's something beyond 5' terminus if you want to work on the 3' UTRs of viral RNAs and things like that. But now I have a question. So your method clearly has a limit of high quality predictions around 125, 130. Beyond that, it goes, like you said, like you have shown on your slides.
Yeah.
Don't just, I mean, getting structures of much bigger RNAs and RNA domains may be difficult because they are not so super common. Many RNA domains are actually relatively small. So my question, what other types of experiments, rather than X-ray crystallography, cryo-EM, to determine 3D structures, could potentially help making better predictions for structures of big domains? Any type of chemical probing, any type of cross-linking? Have you thought about this? Thank you.
Yeah, this is a very good question. And in fact, so I discussed with some of my friends, and we think some cost, I would say the cost information will be helpful. Say if we can input the signal structure determined by some probe or assay, it will be very helpful to again improve the performance of the model. So as you can see here, for this one, puzzle 24. So because in our original version, we didn't consider signal structure. So actually the performance is not very good. So we say if we can input this information, signal structure information into the model and the constraint, say that we ask the final prediction to show the node, will definitely improve the performance.
Yeah, so just a follow-up comment on this. Getting the actual secondary structure may be more complicated, but getting patterns of chemical probing, which are single residue level signals, well, they report different things depending on different chemical probes, could be very easy and cheap to get. So maybe that's a direction.
Yeah, exactly.
Once again, thank you very much. It's a splendid method. We'll try it. My people, I'm sure, have already recorded the links to GitHub. We'll be downloading the method and testing.
Thank you. Yeah, no problem.
Okay, so there is a second question on CASP, so I can maybe build on top of that. So the question is, could you tell us more about CASP results for the proposed model method? And what I wanted to add to this question, what you mentioned is that the biggest problem of the method was purely, or rather on the designed RNAs, that it was difficult to, of course, it's impossible to make alignments, and it was, by human, it was much easier to model. Is there any other reason why the method, was not performing, or like all other deep learning methods are not performing that well for CASP, at least at the moment? What are your thoughts on that?
Okay. So essentially, we try to check the performance and compare the RhoFold performance against the other human expert involved method. So we indeed see a very clear difference regarding the nature RNA's performance and this human designed one. So so as I said, for these ones, their performance are relatively okay. But for this human designed RNA, a number of reasons. So first of all, the patterns actually can be inferred by human beings. So their patterns are kind of biased. And also the RNA sequences here are relatively long. And as I discussed in these slides, so for the longer sequences, for the longer RNAs, the performance is still not very good. And we still need to try to improve the performance for those longer RNAs. Yeah.
Okay, thank you. So maybe now, Arnie, there was a question from.
Maybe these are naive questions, but I'm not an RNA expert, so I ask them anyhow. First, they are related. First, is how did you do the alignments? What the MSAs for RNAs are easy? So that's the question. And related, did you maybe I missed it? Did you try to use a language model as an input instead, like you do in RoFold? Because I mean, it's not that much worse for proteins, and then maybe RNA is even less difference. So have you tried that?
Okay. Very good question. So there are two questions, in fact, right? So the first question is how we construct this multiple sequence alignment, right? So we use a number of tools, including Inferno and also RMSA. Yeah. So for the second one, you mentioned OmegaFold. Do you mean the single sequence based protein structure prediction method? So try to discover
language based models. I don't know. I wouldn't call it a single sequence, but it's I mean, as you did before, you had your language model. So
yes, exactly. So we have already done that, and we are actually working on writing the manuscript. So I can tell you that. Yeah, we have already done that.
How is it performing? Is it much worse? Is it similar?
The performance is in fact very similar to OmegaFold. So compared to the multiple sequence one, it's very fast. So you can get the structure almost in, I would say almost within 10 seconds. So from given the sequence to the final structure, regarding the performance, so it will be a little bit worse, but still, I would say roughly 9 Armstrong if we benchmark using all the PDB structures. Yeah.
Thank you.
Yeah, no problem.
One more question that, sorry, I missed in the Q&A. So yeah, I don't know if Dave wants to say it, but I can also read it. If you need longer sequences.
So this is Jane. I was just wondering why, if you need longer sequences, you don't put in the really good cryo-EM ribosome structures. And, human and E. coli are so different that you don't have much redundancy there. You might have to chop them up because they're too long to deal with. But we found in studying conformation that adding that is just absolutely a game changer. I mean, you get 3 or 4 times as much data.
Yeah. I believe this is a trend. So we can use cryo-EM to resolve the longer sequences with a very high resolution. I would say the recent resolved structures I need of very high quality, and we plan to use them. So we can say all the structures we collected were in last year, May 2022. So at that time, those structures were not available.
But actually,
so,
the earlier 7K00 E. coli is actually way better than the 2 angstroms that it says in its good parts. And so you still get a lot of stuff from there. You know, in the good parts, you can see the holes in 5-membered rings. That's not 2 angstroms.
Yeah. I believe this is a very, very good suggestion. Yeah, I'll definitely consider it. Yeah.
Okay.
Building on that question, do you think it's a problem to mix protein RNA, the data like PDB structures versus just RNA? Because you may imagine that you are trying to predict RNA, but actually in the data set now you have all RNAs and protein RNAs like ribosome, and I think they would behave, because in here you don't really predict proteins. Is that could?
Oh yeah. This is actually another problem. So that is, we try to predict the RNA protein complex, right? So in that case, I would say the kind of different models and all the computational methods are not very capable of doing that, right? So it's kind of you further increase the scale of the problem, right? So nowadays we can predict. We can predict the protein structure or predict the RNA structure, but to predict the complex, not yet that good performance. Yeah.
Okay.
Question. Yeah, 2 more questions here.
So
do you always generate one structure, or is it possible to generate more? Yeah, this is a very good question. So in fact, most of the time we just generate one, but because of this recycling and also the input, so we can, if you want, we can definitely customize the model to try to generate multiple conformation. Yeah. So I mean, when our paper was under review, so the reviewer also raised this question, and we have done some experiment to discuss this. Indeed, so if we really want to generate multiple conformation, we can do that. By default, it's not generated one. Yeah.
Okay, maybe Rachel now, or she wants to close the session because we are running out of time.
Oh, yeah, I have a quick question. Let's see how it goes, and then I'll close the session. So I'll take advantage and take the last question, I guess. So the question is, I guess, regarding a lot of what's been talked about before is artifacts. So I always kind of joke that AlphaFold has learned how crystals, right, the structure of protein in crystals, but for proteins, it seems to be an okay approximation. So my question is, for RNA, it seems we get a lot of situations where it's not an okay approximation, right? It seems like the crystal stabilizing a form that may not be the dominant form in solution. So is there a way in, I guess, in your training regime to still use those crystal structures, but put some information about crystal context or something, so that it can learn where those biases are for each individual training data?
I would say, if we want to do that, probably for the current PDB, database, we need to restructure or try to deposit such information. So nowadays, we just have the 3D structure and the crystal 3D structure, but the information just mentioned is not available. So if it's available, we can definitely try to incorporate such information into the model. Yeah. Obviously, this is a very good suggestion, and we can probably try to see how we can formulate the database construction.